Cape Luther Institute. I'm also a PhD student at Ford International University and the co-PI of the Exuma Sound Ecosystem Research Project here at CEI. I'm mostly focused on reducing negative fisheries interactions with sharks, as well as deep sea exploration.
And I'm Candace Fields. I'm a research technician on the EXO team at CEI, where I mainly help with the shark and deep sea research. And it has been a privilege to teach this class alongside Brendan, and we are extremely proud of all the work they've done this semester.
Hello, I'm Lila Foreman.
Summer Stagman.
Kelsey Taylor II.
Wyatt Huggins.
Fisher Clark.
Lucy Hensley.
Daniel Ayala.
And our research project was characterizing the pelagic fish community in the Exuma Sound. Now, 29% of the Earth's surface is land, and 12.7% of that land is protected. Another 43% of the Earth's surface is high seas, while less than 1% of those seas are fully protected. So clearly, there's a huge gap in marine conservation versus terrestrial conservation, and while in recent years there's been a much bigger push to conserve our oceans, there's still a really far way to go. One initiative that is aiming to further marine conservation is the Caribbean Challenge Initiative, which was an initiative created in 2013 by the United Nations aiming to protect 20% of all coastal marine environments by 2020. And one way that different countries are achieving this initiative is through marine protected areas, or MPAs. MPAs restrict human access to certain areas and put catch limits in place to protect from overfishing. They can be very beneficial in creating more fish, more species, and bigger fish to overall create more productive marine life and more functional food webs in ecosystems. The Bahamas, a country comprised of over 650,000 kilometers squared of ocean, was one country that signed on to this initiative, and as you can see highlighted in yellow are the proposed MPAs that were created to meet this goal. And while this initiative aims to protect 20% of all coastal marine environments, in the Bahamas, coastal marine environments only make up for 27% of its ocean. The other 73% is made up of pelagic water, or open ocean, and this initiative is aiming to protect 0% of those waters.
A problem that exists for pelagic species, specifically sharks worldwide, is that they're highly susceptible to overfishing. Commercial fisheries are moving into pelagic zones due to reduced stocks of fish, as well as improved technology that targets large groups of fish in otherwise desolate environments. Over the years, we've seen a 77% decline in open ocean shark species in the Atlantic, such as the oceanic whitetip and silkie. Much of this is caused by commercial longlining, which is the predominant method for capturing these fish in these areas. Thankfully, longlining without a permit has been banned in the Bahamas since 1993, and because of this, we're able to conduct research in such a pristine environment, and we can gain knowledge about what a healthy pelagic ecosystem should look like. To do this, we're working under a project called the Exuma Sound Ecosystem Research Project, or EXERP, which seeks to understand open ocean habitats off of Cape Eleuthera. Under EXERP, we use longlining, fish aggregation devices, and transects, where we've actually had some encounters with Blainville's beaked whales and Risso's dolphins. Although all of these are important, our project is focusing on the use of longlining with the goal of characterizing the pelagic fish community in the Exuma Sound from 0 to 400 meters.
In order to meet this objective, we set up longlines in these 3 different fishing sites shown here on this map of Cape Eleuthera, where we all are currently, overlaid by a depth chart. What's interesting about these 3 different fishing sites are they're all equal distances from the wall, which is an underwater shelf that drops from around 20 meters deep to over 1,000 meters deep. They're also all very close to our base station here at CEI, indicated by a star, and they're all different depths. And so we set up long lines at these different sites at different times of the day and night to see what we can catch and when in the pelagic ecosystem of the Exuma Sound. And then again, we use our long lines to survey the pelagic fish community from 0 to 400 meters deep. We use 3 hook sizes for our long lines and 2 leader types for our gantries. Gantries being the ropes that the hooks hang off of the long line from. We use monofilament leader and braided wire leader. We use 2 different bait types for our long lines as well. We make use of mullet and squid, and again, this is to maximize the opportunity we have to catch a wide range of pelagic predators in the Exuma Sound. Here you can see a diagram of a long line. It's essentially a long line stretched between buoys that hang suspended in the ocean. Here you can see the gangents to which the hook and the bait are attached, and the fish will be attached to the long line once it bites onto the bait until the line is hauled in.
After we set the long line, we wait about 4 to 5 hours to haul the line. When we catch a fish, the first step is to secure it on the side of the boat, where we then take measurements. These measurements help us to identify what the fish's maturity. And after we take measurements, we take a fin clip. This is so we have a DNA sample of the fish. We then check for parasites, and we check the sex of the fish. We then tag the fish, and in the future we hope to use satellite tags in the study. This is so we have a better understanding of where these pelagic fish go after we release them. We then take blood and muscle samples. This is so we have an understanding of what the fish has been eating over the course of its life, so we have a better understanding of where it lies in the food web. These samples go to CEI, where they can be used for other research projects, not just solely our research project.
We will now be giving a bit of information on the fish which we have captured. You can disregard the majority of the information on this slide. For now, it will be covered individually momentarily. The main purpose of this is to show that we have caught 6 fish, despite putting in over 3,700 hook hours, which is the combined amount of time which all of our hooks have spent in the water. Now, this may seem like a very low catch rate compared to the effort put in, but this is to be expected in the near desert-like environment in which we are fishing. Something else we have considered is the vulnerability of the 4 species we have captured. The International Union for the Conservation of Nature, IUCN, has created a scale ranging from least concern to extinct in regards to the population stability of species globally. There are 3 points on the scale considered threatened, those being vulnerable, endangered, and critically endangered. One fish which we have captured so far this semester has been the tiger shark. We caught one. It was 179 centimeters, or a little over 5 feet. It was an immature male, and the global populations of this species are near threatened. Another fish we have captured so far this semester, in fact the only one which does not happen to be a shark, is a snake mackerel, which, despite what the name might suggest, is not considered a true mackerel. Its size was 70 centimeters, and it was an immature female. This species is a prime example of a vertically migrating fish, which is to say that during the night they move up in the water column from the deep depths that they normally inhabit to follow their main food sources, which include small fish and squid. The global populations of this fish are least concerned. The most commonly caught fish so far this semester has been the silky shark, which has accounted for half of all of our captures. The mean size of them were 112 centimeters, or a little over 3 feet. And the at-best mortality rate for this fish was 33%, meaning that one of them did unfortunately die despite our best efforts to keep the fish that we capture in good health. They were all immature males, and the global populations of this species are vulnerable primarily due to their accidental capture on commercial long lines.
The most amazing and unusual fish that we have caught this semester has been the oceanic white-tipped shark. It measured 92 centimeters in length, or a little under 3 feet, and was released in good condition. The oceanic white-tipped shark is considered critically endangered in the Atlantic by the IUCN's Red List and is considered vulnerable in global populations. Specifically, we have seen an over 95% decline in these sharks' populations in the Atlantic in the past few decades. CEI has done extensive research on oceanic white-tipped sharks in the Bahamas, specifically off of the southern tip of Cat Island. And this CEI-led research team has tagged over 100 individuals with satellite tags, many of which were confirmed pregnant females. However, these satellite tags have still yet to show us where these females go to give birth. In fact, they have never tracked an oceanic white-tipped shark into Exuma Sound until this semester. We are the first group of people to catch an oceanic white-tipped shark in Exuma Sound, and not only is this the first of its species to be tracked into this area, but it is also a juvenile oceanic white-tipped shark, which hasn't been caught in nearly a decade of research in the Bahamas. What is so spectacular about this specific shark is its age. This graph by Sicki et al. 1998 compares the precaudal length in centimeters, which is from the head to the base of the tail, to the age in years. If you follow the curve, you can see that the newborn oceanic white-tipped shark's precaudal length is 60 centimeters in length. And the shark that we caught was only 63 centimeters in length. This means that the shark was under 6 months old and was probably born in the Bahamas. This means we can finally start answering the question, where do these females go to give birth? And here's the shark swimming away from the boat after being worked up. This is honestly one of the coolest fishes in the ocean, and we are truly lucky to have experienced it this semester.
Because of their low reproductive rates, pelagic predators are not sustainable to fish. Over the course of the past 3 months that this study has taken place, we have already gathered a lot of valuable information. Very little is known about the pelagic region, but we have already discovered the importance of the Exuma Sound to various different species. We've caught fish that have never before been seen in this area, which confirms our belief that there's a lot to learn about this region. Recently, there have been talks about opening up pelagic species to commercial fishing, which would likely mean lifting the longline ban. Based on our catch rates, not only would this be unsustainable, but it would also not be economically viable, as we spent a lot of time and money setting longlines and only caught 6 fish, none of these being game fish, which would be the target of these commercial fisheries. Not only this, but also longlining would be detrimental to shark and other pelagic species populations, as they are small and easily overexploited. Before the Bahamas or other Caribbean nations can begin to think about protecting their marine territories and meeting international commitments such as the Caribbean Challenge, we must know what species occur in the open ocean and what their movements are. Although our data set is limited, this study will continue into the future, and we expect catch rates to increase in the coming months. After deploying satellite tags to track animal movement and collecting more samples to add to our data set, we will have a better picture of the pelagic ecosystem as a whole and ultimately can inform their conservation. Here's some literature that we cited, and I'd like to give a big thanks to Brendan Talwar and Candace Fields for being amazing research advisors, and I'd like to thank Juliet, Jessica, Emmy, Lily, and Jack for being a great part of the intern team. And I'd like to thank the FATS communication and kitchen team for always helping us out. Thank you.